Well, hello, and this is an attempt to do the full Chapter 9 voiceover on these PowerPoints. I know that it will be a long um, process, but again, if you do things correctly, I believe we can break it up. You can listen to it at different segments, um, but as I'm breaking it down, trying to do it in individual segments, it's just taken way too long. So this is my attempt. So to begin with, there are three types of muscles: skeletal muscle, cardiac muscle, and smooth muscle. Skeletal muscle is the only voluntarily controlled muscle, and one of the only voluntarily controlled things in the entire body. In general, it attaches to the bones of the skeleton. There is one little area of exception in the muscles in the face, but that's a rarity. Cardiac muscle is obviously in the heart. It is involuntary. Skeletal and cardiac muscles are both considered striated muscles because they appear to have striations under the microscope. Whereas smooth muscle is called smooth muscle because it does not have those striations. But cardiac and smooth muscle are both involuntary, which means you have no conscious control over it, that it is controlled under your conscious effort, below your conscious effort. The other thing about cardiac muscle that I do want you to know, and we'll look at this later on, is that they have what are called intercalated discs, which are simply the little desmosomes, the little spot welded areas from one of the earlier chapters that we talked about, where the cells are really held together firmly. And it's because cardiac muscle does not have the luxury that skeletal muscle has of having some kind of really strong anchor on either side. Cardiac muscle has to pull on its own. So skeletal, cardiac, smooth. Skeletal is voluntary. The other two are involuntary. Skeletal, cardiac are considered striated, where smooth is non-striated. So skeletal muscle, again, like we said, attaches to bones, except in the one rare instance when it attaches to the skin and the face. These are called the muscles of facial expression. And again, all these muscles are under conscious control. They're voluntary. And it is important to understand that every muscle is an organ in and of itself. So because they have multiple types of tissue, like it says underneath. Now, muscles are going to have a special connective tissue that's going to be associated with it. One of them is called fascia. Fascia is simply the outer, really tough layer of connective tissue that covers each muscle. It keeps them separate from each other. Again, in general, if you've ever eaten a steak and experienced something that most people commonly call gristle, that's what fascia is. Now, the other one is going to be tendons. Those two are pretty common. Tendons are the connections that muscles have to bones. And so anything where it's a muscle that's going to be connected to a bone, this is where we call it a tendon. Now, don't forget, ligaments are where bones are linked together. And then the third type of connective tissue is a very rare one. It's called an aponeurosis. There's really only two. You have one between the frontalis and the occipitalis in the cranial area. And then in the rectus abdominis, you have this aponeurosis between the different pockets of muscle. And all an aponeurosis is instead of like a tendon has a muscle on one side and a bone on the other, an aponeurosis has muscle on either side of it. Then we have the connective tissue covering of the actual internal structures. So outside of having the fascia around the outside, we also have a thin layer called the epimyosin around the outside. It is just underneath or deep to the fascia. If we were to cut the muscle in half and look at it, you would see a bunch of segments, little tubes. These tubes are called fascicles. Each fascicle is surrounded by perimyosin that separates the fascicles from each other. And then within the fascicle, if I was able to enlarge it like in this picture and cut it in half and start looking at it, you would see it as well has all of these tubes in it. Each one of those tubes is an actual muscle cell, which can also be referred to as a muscle fiber. And so they are each surrounded by endomyosin. So endomyosin keeps all of the muscle cells separate from each other inside the fascicle. It really is like this little repeating pattern. It's like one of those little Russian, I think they call it nesting dolls or whatever, where you open it up and there's a little doll that looks almost the same underneath, and you open it up and there's another little doll. Because even inside the muscle fiber itself, we're going to find repeating patterns of these tubes. We're going to call those myofibrils. And then inside the myofibrils, if I was to cut it in half, you'd see little tubes that we call filaments. But on this page, remember, we're looking at epimyosin, paramyosin, and endomyosin. Epimyosin surrounds the whole muscle. Inside the whole muscle, there are bundles called fascicles. They're surrounded by perimyosin. Inside those fascicles are muscle cells, which are also called muscle fibers, and they're surrounded by endomyosin. Now, this picture is going over the inner structure of a muscle cell or a muscle fiber. Now, there's a lot of special things in here. Remember that muscle cells do not have a normal cell membrane. They have a lot of special things on it, and so the muscle cell membrane is called a muscle fiber, or is called a sarcolemma. I am so sorry, I just saw that up at the top. So sarcolemma is the name for the muscle cell membrane. And the sarcolemma is designed to transmit an electrical stimulus across the entire muscle cell. And so as it transmits this, there's going to be these little tubes that extend inward that are basically just extensions of the sarcolemma going inside the cell, and they're called transverse tubules. Now, on this cross section, you can see the myofibrils, these tubes inside of it, and the transverse tubules are kind of pointed out. They're these little kind of yellowish rings that wrap around them. Now, in between the transverse tubules are this little webbing called sarcoplasmic reticulum. We're going to learn that these actually hold and store calcium in just a little bit, because calcium is the key to muscle contraction. So inside the cell, we have these repeating structures called myofibrils. The myofibrils are going to have a webbing called sarcoplasmic reticulum that wrap around them, which is going to hold calcium. And at intermittent stages, you're going to have these tubes called transverse tubules, sometimes called T-tubules, which are simply extensions of this sarcolemma, this special cell membrane that muscle cells will have. Now, each one of these myofibrils has segments in them. So if I was looking at a brick wall, each brick would be a segment inside the whole wall. So inside the whole myofibril, it's segmented out, and these segments are called sarcomeres. Now, the boundary lines for sarcomeres are things called Z-lines, sometimes referred to as Z-discs. And that's the boundary. From Z-line to Z-line is what's called a sarcomere. And now these sarcomeres are the actual fundamental unit of contraction. They are the part of this myofibril, this whole muscle system that is actually doing the machinery and the work of contraction. Inside the myofibril, or inside the sarcomeres, which make up these myofibrils, are different filaments. There's going to be a thin filament called actin and a thick filament called myosin. The thin filaments called actin are attached to the Z-lines. The thick filaments called myosin are in the middle. And what's going to happen, we're going to look at this, that the thick filaments, these myosin filaments, they are going to reach up and grab the actin on either side of it and pull it towards the middle. And so if we look at a sarcomere and look at the way it's made up, we can see the thin and thick filaments form these dark and light and medium stripes. This is where striations appear. Right? That's why skeletal muscle is called striated, because skeletal muscle and cardiac muscle have sarcomeres. Smooth muscle does not. It has a different kind of arrangement. So inside this sarcomere, if I look at it, I'm going to have a middle medium band, and it's called the H zone. And then on either side of that, I'm going to have these darker bands. And in this picture, they're purple. They're not actually named, but the edge of the purple on either side, the boundaries of it, make up what's called the A band. All right, so the H zone is simply the area where the myosin is not covered up, and the A band is just the length of the myosin. So on either side of that, you're going to have this really light band that's going to cross over from one sarcomere to the other, and it's called an I band. The I band is the area where the actin is uncovered. So as the myosin, this A band, starts pulling the actin towards the middle, which is called the M line, the little area called the I band and also the H zone will get smaller, because there will be less and less of each one of those, myosin or actin, that will be uncovered. It's like pulling your bed sheets up over you or whatever. So there are 5 areas that you need to know. The I band, which is simply the uncovered actin. The A band, which is the length of the myosin. The H zone, which is the area of uncovered myosin. The Z line, which is the boundaries on either side. And then the M line, which is the middle line. This gives another kind of look to it. If you've got Connect, I believe there's some videos that you can see in there that show this contraction. I will try to find some and upload them for you. Now, this is where it's going to get important to kind of see some structures. So inside the sarcomere, again, we talked about the thick filament being myosin, the thin filament being actin. You can kind of remember that the thin filament is actin because if you are active, generally you'll be thin. Now, that's not a total always situation, but it's a good rule of thumb. So the actin is the thin filament. Now, myosin's pretty straightforward. It's going to have these little parts of it that stick out almost like Velcro. They're going to have these little things called myosin heads, and they are what is going to try to bind with axin to form these cross bridges. But these myosin heads stick up at an angle. The angle is pointed away from the M line and towards the Z line when these are not bound to each other. The actin, the thin filament, this has got 2 really important proteins to know. One protein is called troponin, and the other one is called tropomyosin. Now, the tropomyosin is the one that's going to prevent the binding, and that's why it has myosin in the name of it. It is going to get in the middle of actin and myosin. Now, on the actin, there's also another little thing called the binding site. On this picture, you can kind of see it. If you imagine that the actin looks like 2 twisted rows of martini olives, I kind of look at it as the little area where the pimento is. That little opening, that would be the binding site, and that's what tropomyosin is going to cover. And again, you can remember it's tropomyosin because it has the myosin name in it. It is trying to get in between myosin and actin. Now, it is held together by troponin. Now, troponin is simply holding the tropomyosin over the binding sites, right? So that there's a little mechanism that's going to happen that's going to unlock, and a couple of stages will occur in order to cause a muscle to contract. But the first and foremost thing is to know where things are, which things are on which one of these filaments, and what they do. So remember, myosin doesn't have much except for these little cross bridges, these myosin heads. On the actins, where most of the structures are, we've got a binding site that's covered by tropomyosin, and then tropomyosin is being held in place by troponin. It's really important to know these, because once we start looking at how muscles contract, if you don't have a good understanding of the anatomy, what the structure is, as we start looking at how it works, the physiology will be kind of tough. So contraction of a muscle fiber, there's a lot of different things that are going on. As it says on this, it requires interaction of several chemical and cellular components. But what's going to happen is each sarcomere is going to shorten, and since there's thousands of them along a myofibril, the whole myofibril shortens, which means the whole muscle is going to shorten, because there's several muscle fibers in each muscle. Now, what triggers this is a signal from the nervous system. Again, in the skeletal muscle, it is a consciously controlled signal that is going to come from your brain down to a muscle to cause it to contract. Now, wherever an axon ends is referred to generically as a synapse. Now, that's a word that will be used for most of the places that an axon ends, but in this instance, this synapse has a special name. It's called a neuromuscular junction, where the neuron and the muscle join. That's a well-named area. So you'll see that most of the things on here are going to have synapse in the name, but first of all, just remember that it is a neuromuscular junction. So, to start with, when I decide to contract a muscle, it is a motor neuron that is going to send the signal down to that muscle. Now, we have two basic types of neurons in the peripheral nervous system. We'll look at this in the upcoming chapter. You have sensory neurons, which bring information in, and a motor neuron, which take information out. And when I say in and out, I'm talking about to the central nervous system. So sensory brings information into the central nervous system. It's called sensory because I'm sensing my surroundings. Whereas motor is called motor because it's going to be going out and causing a movement, something to happen. So where this neuromuscular junction is, it's where a motor neuron, the axon of a motor neuron, comes in contact with a skeletal muscle. And just to make sure I don't mislead anybody, there's no direct contact. There's actually a little space between them as it fits in. And if you can see the little inset of the picture, you can see there's a little area. It's referred to as the synaptic cleft. All right, so the motor neuron comes in. It will have a bulb, a synaptic bulb they're called, at the end of an axon. And it is going to fit into a little indentation inside the sarcolemma. Again, it won't touch it. It fits into a little groove, so there'll be a synaptic cleft between the motor neuron and the sarcolemma. Now, the area of the sarcolemma that's directly underneath this synaptic bulb is called a motor end plate. The motor end plate is where this chemical we're going to talk about in a minute is going to bind, and it's going to cause an electrical current to be created. Now, whenever I stimulate this and I send a signal on a motor neuron down to a muscle, once it gets there, there will be a release of a chemical that is in general referred to as a neurotransmitter. Right? That's how neurons send their signal. It's a neurotransmitter that is released. Now, in skeletal muscles, that neurotransmitter is always, and I repeat, always acetylcholine. Now, let's look at the next slide. So the next slide is about acetylcholine. Acetylcholine is the neurotransmitter that is used in all skeletal muscle contractions. Neurotransmitter is the most common type of neurotransmitter in the body. We're going to see that it's used in the majority of things in the central nervous system and all skeletal muscles. So if you ever have a question about a neurotransmitter and you don't know what to answer it, guess acetylcholine. So Once this acetylcholine is released, this neurotransmitter is released, it's going to bind to receptors on the motor end plate, that little specialized sarcolemma just below the synaptic bulb. Once it does that, there's going to be a change in the membrane potential, the ions that are on either side of it, causing an electrical charge to be passed along the sarcolemma. Now, once this electrical charge we were just talking about has been passed along the sarcolemma, it is going to go in through the transverse tubules and then will electrify basically the sarcoplasmic reticulum, causing the calcium ions to be released. And calcium ions are the key to muscle contraction. Now, last chapter we talked about the regulation of blood calcium levels. This is one of the big reasons why your body or our body will raid our skeletal system to get that calcium that's in there if we are low, because muscles need to contract, especially the cardiac muscles. So I'm going to have a motor neuron that's going to send a signal into the skeletal muscle. It's going to drop off acetylcholine, which is going to bind to receptors on the motor end plate. It's going to cause an electrical current to be created that's going to go throughout the sarcolemma, the specialized skeletal muscle cell membrane. It's going to take that, the sarcolemma will take that message, and then it will go into the muscle cell itself by way of transverse tubules. And then the transverse tubules will electrify and basically cattle prod the sarcoplasmic reticulum that's next to it, causing that sarcoplasmic reticulum to release the calcium ions. Now, we're going to look at how this works, but we have to remember what the anatomy was of the actin and myosin. Remember, actin has the binding sites on it. The binding sites are covered by tropomyosin. The tropomyosin is held in place by troponin. So this is where we're going to look at how this happens. This page shows a cycle of a muscle contraction. Now, if you look at the top right corner, you will see an extra thing that wasn't talked about. And it is that on the myosin heads, while the myosin heads are relaxed, there is going to be two chemicals. One's called ADP and the other one is called phosphate. Now, if I was to get both of those to bind together, it would form a molecule called ATP. ATP stands for adenosine triphosphate, where ADP is a diphosphate. So there's three when it's ATP, only two phosphates when it's ADP. So that first down arrow underneath that is showing that calcium's been released. So these are the steps of the muscle contraction. Calcium gets released, it is going to bind to troponin. Troponin is going to let go of tropomyosin. Tropomyosin will then roll off or uncover the binding sites. Now, once the binding sites are open, the myosin heads are going to want to bind with them. That is their desired little thing. So the binding sites, once they open up, the myosin heads will bind with them. Now, myosin heads can only hold on to one thing at a time. And so they will let go of the ADP and phosphate. And when they do, there is an actual change in the molecular structure right at that bend. Now, remember, they have just bound to actin. When they let go of ADP and phosphate, that myosin head will bend from being pointed out toward the Z line to being pulled in toward the M line. And because they're bound to actin on either side, they're going to pull actin towards the M line or towards the middle. Therefore, the sarcomere will start to shorten. This is referred to as the power stroke. Now, Once ADP and phosphate have been released from the myosin heads, they will bind together and form ATP, which is the myosin head's most desired thing. So the ADP and phosphate, once it gets together and forms ATP, the myosin heads look at that and they're like, all right, it's on. So it grabs the ATP, and in doing so, because again, it can only hold on to one thing at a time, it lets go of the binding sites. Now, like a kind of a cruel joke, as soon as it lets go of the binding sites and holds on to the ATP will break back down into ADP and phosphate. And so it'll set back up into its original position. It will go back out pointing, the myosin heads will be pointing outward towards the Z lines. So as this happens, and this would happen at, just a fraction of a second. This is going to go on. This is going to happen hundreds, if not thousands of times in a second. As long as acetylcholine is still in the receptor, then the calcium will still be released and it'll still be bound to tropamine. Therefore, the tropamine had let go of the tropomyosin, so the binding sites are open. So this little cycle is going to keep going on until our body decides to quit contraction, and in that instant, it will release something called acetylcholine esterase. We'll talk about that in just a second, but I always mispronounce it because I like to say acetylcholine erase, because it really is going to erase the acetylcholine. But once that happens, once acetylcholine esterase gets released, then the calcium, the electrical signal will stop. The T tubules will no longer be electrifying the sarcoplasmic reticulum, and like a big hoover, the sarcoplasmic reticulum is going to suck back up the calcium, and then everything will set up back where it was, right? Now, this entire thing that I just went over on the previous slide is called the sliding filament model or sliding filament theory. I will try to put up several videos online if I can that will go over the sliding filament theory so you can see it in action, and the Connect has it as well. What's going to happen as that does that, or let me stop and just say, if you look on YouTube, make sure you look at reputable sites. There will be some ones that aren't really all that good. But anyway, so as this happens, as the muscles contract, the sarcomeres are going to shorten because the myosin is pulling the actin towards the middle. And since the actin is attached to the Z lines get pulled closer towards the middle. And so this is how muscles contract. Now, as we talk about this, I just went over, and I'm not going to kind of go over this one more time, but we'll talk about it again in a minute. But just remember, it's called cross-bridge cycling, as it binds and pulls and lets go, and then binds and pulls and lets go. But it is a pretty solid fact that that's how muscles contract. So now we talk about acetylcholine esterase. Again, I mispronounce it on purpose, acetylcholine erase, simply because I like to remember that it erases acetylcholine. This is an enzyme that when it's time to stop muscle contraction, this enzyme is released into the synapse and it breaks down acetylcholine and stops the impulse. It's all pretty much instantaneous. Now, there's a calcium pump that's going to pull the calcium back into the sarcoplasmic reticulum. The tropomyosin complex goes back to where it was. So then the muscle fiber starts to relax. So when we look at the source of energy, where they come from, there's actually three types. Now, ATP is stored in small amounts inside your muscle cells. This is used for everyday stuff. If you're writing something down, there is muscle contractions going on. If you are, simply walking into the kitchen or whatever, you're going to use an amount of ATP. Our body is constantly monitoring the energy output that it goes through. And so the energy output is going to be measured, and my body will have ATP for my base level activity. If I need to do something like, let's say, I decide I need to get up, I see the the mailman come through, and I don't want to go to the post office, and I've got something I need to mail out, and I need to run outside to catch him, and I have to sprint for like, I don't know, 30 seconds or a minute, 30 seconds probably. Anyway, during that time, that's going to take up the ATP reserves, but we'll have this compound called creatine phosphate that's in our cells, and that's designed for short bursts of energy. It's really not there for anything major. It's there for about what we'd say maybe a 4-minute burst of energy, something I wasn't planning to do, but I need to do. But once it gets past there, then we go through what we call cellular respiration. Now, we looked at this in chapter 4, but we are going to go over it again. All right, so we've got ATP in reserve for normal baseline movements. If I have burst energy where I have to do something for a brief period of time, a few minutes, that's going to require more, then I tap into my body's creatine phosphate reserves. Once I get past that, then I go into cellular respiration, which is the breakdown of glucose. Now, cellular respiration is going to happen in 2 phases. There are 3 steps in those 2 phases. The very first phase is called anaerobic, which simply means without oxygen. Once glucose enters into the cell, it is now in the cytoplasm. It will break in half. Glycolysis, glycolysis, is a word that means, lysis means to break down, gluco means glucose. It literally means to break in half. And so this first stage where glucose, the molecule, enters into the cell and gets broken in half is called glycolysis. It does not require oxygen, so it is referred to as an anaerobic phase. Now, as this glucose is broken down, it will be converted into what's called pyruvic acid. Now, we're not going to get into all the major steps, but this pyruvic acid, if oxygen is present, will then be changed and taken into the mitochondria. The mitochondria is where the next 2 steps occur, and since it requires oxygen to get there, it is referred to as the aerobic phase. So it is going to produce a lot of ATP. The 2 phases are the citric acid cycle and the electron transport chain. Now, the electron transport chain is where the majority of ATP is going to be produced. But the main thing here is the aerobic phase, which requires oxygen, happens inside the mitochondria. And it is 2 steps that will occur to have this happen. So cellular respiration is going to have 3 steps: glycolysis, citric acid, electron transport chain. Glycolysis happens inside the cell as soon as glucose enters, and it does not require oxygen, so it is referred to as an anaerobic phase. If oxygen is present, the pyruvic acid gets taken inside the mitochondria where the other 2 steps happen. So these are referred to as aerobic phases. And again, these occur in the mitochondria, and this is where ATP is produced. This is why if you run, if I haven't been to the gym in forever and I go and I get on the treadmill, I go through a minute or 2 and all of a sudden I'm going to be breathing heavy because I can't process oxygen efficiently enough, right? So the aerobic phase is literally your cardiovascular health, right? Now, as it says here, and unfortunately the book doesn't do a great job of kind of pointing this out except for this one little part, that there's an extra special type of molecule found in muscles called myoglobin. It is a cousin to hemoglobin. Myoglobin, myo means muscle, hemo means blood. So myoglobin is hemoglobin, but it's inside the muscle cells, and these are going to be there to store oxygen so that we can make sure we can get into the aerobic phase. Now, here's the big thing. When you exercise and you do things to kind of tap into the cellular respiration to get your body used to it, this will increase the amount of myoglobin in each of the cells. So in exercising, it gets your body to produce more myoglobin. Now, here's another thing going back to the conversion of this pyruvic acid. If there is no oxygen present, pyruvic acid then gets converted into lactic acid. And most of us have probably heard of lactic acid. Lactic acid is the byproduct if I work out too much, or and I haven't worked out past my point of endurance, and my muscles will get sore. It's because of the lactic acid that builds up in it. Now, this is going to lead us into the lactic acid is going to lead us into this next slide. So, when we are exercising, like let's say we haven't gone to the gym in a while and we decide, you know what, it's time to go back. And we start on the treadmill and, you start looking and you think, man, yeah, I used to be able to run, marathons and it's all great. And you get on there and after like 3 minutes, you're huffing and puffing and you're like, wow. What's going on is you've gotten past your body's ability to use oxygen. But once that happens, your body still will let you burn energy. And it's going to give you basically a credit card. So that bottom one on this page that talks about oxygen debt, that's what's happening. I'm going past my ability to use oxygen efficiently, so I'm just going to start burning glucose at a higher rate. But that's going to be where my body's going to say, hey, it is, once I stop exercising, I get off that treadmill, I'm going to start breathing really heavy. I'm going to have a hard time. My body's saying, you need to repay back that oxygen. You need to repay back that oxygen that you used. And one of the other things that's going to happen, like it says above, here the anaerobic threshold or lactic acid threshold, during stimulus muscle activity, so I'm on the treadmill and I'm not used to it, I'm going to start making lactic acid. That's why I'm going to be sore the next day. Now, what's going to happen is a lot of that lactic acid is going to be taken into the liver. And at the liver, it's going to wait for the oxygen to come through. And the liver is one of the only areas in the body that will take that lactic acid and convert it back to glucose. The only other area that can do that is the only muscle that is always working. The heart has the special ability to take the garbage of overwork, overstress called lactic acid and convert it into energy called glucose. So the liver and the heart are the only two areas that really can do that, which I find pretty amazing. Now, this picture is kind of going through, we talked about this in chapter 4, I believe, where we talked about how our body stores glucose as glycogen. So after we've eaten, as our liver is digesting stuff, it's going to be taking it through and it's going to put it in, it's going to change it and put it into a new form where it combines hundreds of glucose molecules together in this big molecule called glycogen. So the arrow up from glucose to glycogen shows storage, but the arrow down from glycogen to glucose is where I'm going to be needing it for energy. And so this is kind of the cycle as it goes through. Now, this is one of the slides that just makes me laugh. I don't know how this got put together, but now look, there is a time of muscle fatigue. I don't know how many people have ever experienced it. If you've ever been involved in athletics, you probably have. I have, personally, where it is where you work out so hard that your muscles just say they're not doing anything anymore. I used to have a stick shift when I was in college, and early in college, my car didn't even have power steering. I'm sure there's a lot of you that don't even realize that not all cars used to had power steering. And if you've ever tried to drive a car that doesn't have power steering, when it is not moving, it is almost impossible to turn the wheels. But there were times when I would get out of my sports and I would sit in my car and I literally couldn't drive because my arms were jello. I went through muscle fatigue. There's a lot of things that are going to be on there, and I will tell you the third one on this list where it says loss of desire to continue exercise is just stupid. That is one of the dumbest things I've ever seen on one of these slides. The loss of desire to exercise is not muscle fatigue, that's brain fatigue. All right, and if you've ever experienced muscle fatigue, you'll know that it wasn't, that you probably wanted to quit a long time before that. And the other thing is, I don't know why they have this as controversial, that it's an accumulation of lactic acid, because that's what's going on. Look, lactic acid, in case you couldn't figure it out, is an acid, right? Acids denature proteins. Muscles are entirely protein. That's why if you're eating a steak, it's a protein, right? If I put too much lactic acid in there, it will do damage that won't be able to be reversed. And so our body has built-in mechanisms to stop us from over-exercising and putting too much acid inside the muscles to cause serious damage. Now, muscle cramps, now we could get into all kinds of different things about muscle cramps. The only thing that I want you to know in this book, we'll talk a little bit about them, another generic part later on, just the basic name of them. But in here, all I want you to know is that it's a sustained involuntary muscle contraction. It can be caused by all kinds of different things. There are all kinds of different things that cause cramps. But all I want you to know is it is a sustained involuntary muscle contraction. Now, in the act of contracting a muscle, over half of all the energy is released as heat. So in all the power that is needed to cause a muscle to contract, the majority of it doesn't go to the force of contraction, it goes to heat. And so this is why if you go to the gym, it feels like it gets hot. If you start doing something active, your temperature will start raising. Now, this is a really important thing, just to kind of as a byproduct. This is also why if you ever have the flu and you have a fever and your legs, your thighs, and your back get sore, you don't realize it, but your body is sending signals to cause those muscles to contract to help bump up your temperature. So as we start measuring how things work, we're getting ready to look at how it's an electrical device that they use to show a single muscle fiber contracting and measure things on it. So when we take a single muscle fiber and cause it to contract, we can measure how much energy is needed to cause it to contract, and we call that the threshold stimulus. What is the boundary of an electrical impulse that it finally gets to that causes the muscle to go through the contraction process? And so one action potential from a motor neuron releases enough acetylcholine to produce threshold stimulus in a muscle fiber, causing the muscles to contract. This contraction is called a twitch. And so if we looked at it on this little graph and we looked at a twitch, we would see that there's three parts to it. And this is a very short, these little units of time are very short. The first part is called the latent period. The latent period is simply the time it takes from the acetylcholine to bind to the receptors until that first junction of the myosin heads and the binding sites where it pulls it forward. So the first act of motion. So there's a point in time. Acetylcholine is released, bound to the receptors. It causes an electrical impulse on the sarcolemma that gets taken into the cell by the transverse tubules. It electrifies the sarcoplasmic reticulum, which lets go of the calcium. The calcium binds to troponin. Troponin lets go of tropomyosin. Binding sites are exposed. The myosin head binds to it and pulls towards the middle. All of that is called the latent period. And basically, it's simply saying there's no movement going on. We've got stuff happening, but it's not shortening. Then we go into the period of contraction. The period of contraction is as the muscle is shortening. Now, there is a point in time when you say, all right, I'm done with it. After that, and the acetylcholine esterase has been released, and it dissolves the acetylcholine, and everything starts going back, we go through a period of relaxation. So there's three parts to a twitch. Latent period, where no movement is happening. Contraction, where everything is being pulled towards the middle. Relaxation, where things are going back to where they were. Now, the way that a muscle fiber contracts is an all or none property. We're going to look in a little bit at how muscles are used and regulate the amount of muscle fibers that are contracted to control the strength. But we can also control the strength by the overlap. And if you look at this little diagram, and I'm going to start off to the right, let's say where it says overly stretched. When I stimulate this muscle to contract, you should be able to see that there's a whole section of the myosin heads that aren't covered over. And so when we start trying to contract, it's like saying I'm trying to do a tug of war, but if I have 10 people on each side, instead of all 10 pulling, I'm only using about 3. It's not going to have as much force. And so I'm wasting some effort. But once it starts pulling over, it'll get to this optimal length right in the middle, where all of the myosin heads are grabbing onto the actin. At that point, we're going to have the best possible contraction. Now, once the contraction happens, it will get to where where the actin starts bumping into the other actin in the middle, and that's where it will quickly diminish the strength of the contraction. So if we look at how these strengths of the contraction and plot them out, these twitches and the strengths, what we can see is if I stimulate, like on this top one, the graph A, if I stimulate the muscle to contract and I have it go up, it goes through the contraction period, and I have it go back to the relaxation period, and I let it relax all the way back down to where it was, and then stimulate it again, I'm going to have the same force. But if I stimulate the contraction like on this middle one, and we have the period of contraction, and then during the period of relaxation, if I stimulate it again before it gets back to its original position, I will probably have better overlap. And so this contraction is going to have a stronger pull. And I can do this, and it is a very scientific process that we know very well now, but this is called summation. And it is what a lot of athletes do that are in power competitions. It's kind of like warming up, but it's a very regimented way of warming up to produce a stronger and stronger contraction. And then the bottom one is called a tetanic contraction, which is a cramp. Going back to what cramps were, we said that they were a sustained, uncontrolled muscle contraction. I want you to know that the name of a contraction, a cramp, is a tetanic contraction. That's where we get tetanus, lockjaw, right? Now, our body can use what's called motor units in order to control how much force in each given muscle is used to do something. So our muscles are divided up into segments called motor units. All a motor unit is one motor neuron and all of the muscle fibers it stimulates. So we can have one motor neuron that will have hundreds of muscle fibers. That would be in something like a large thing like the gluteus maximus, or one of our larger muscles. Or a motor unit can only have a dozen or so muscle fibers in it, like the small muscles that work the eye. But the one thing it has in common is that it's one motor neuron. All right, so our body can divide things up so that if I'm going to pick up, like let's say I'm drinking my coffee and I'm drinking water now. When I bring this up to my mouth, I'm not needing my whole bicep brachii to contract to get it to my mouth, right? So, but if I wanted to move a box of books, I would use my whole bicep brachii. So I can regulate the number of motor units in a contraction depending on how heavy an object is. Now, some of you might have experienced where our brain is monitoring kind of the idea of the weight of something. If you've ever picked up something that was heavier than you thought it was, or on the other side, if it's lighter than you thought it was. If it's heavier than you thought it was, you probably didn't have enough motor units and you can hurt yourself. And you can hurt yourself if it's lighter than you thought it was. You have too many motor units and jerk it up. But that's how it works. The other thing is called recruitment, right? So my body will use different amounts of motor units in order to cause a smooth movement. Now, this is an area where you won't see it until we go past its efficiency. So when I was in school, I lived on the 4th floor. And if you live on the 4th floor of an apartment that doesn't have an elevator, it becomes a real challenge in how you shop. And so I would go, you either become a pack mule, you go and you do one big shopping thing, or you do multiple little shopping things. So I would just load myself up and go up to the top floor. And by the time I got to the top floor where I was, it was hard for me to get the key in the lock because I would be shaking so much. This is what happens. Our body will use a certain amount of motor units in a muscle. And when they fatigue, if I've been using that muscle for a period of time, as they fatigue, our body will rest those and recruit others. And it does it in a smooth transition so you don't even know it. But if I go past the ability to find a rested motor unit, then the muscle will start shaking and it will look like tremors because I don't have a rested one to use and my body's trying to spin things around to find somewhere. Hopefully that makes sense, but that's how recruitment works. Now, when we talk about these recruitments, our body is going to, as we fatigue, even if it's light, we will start recruiting larger and larger motor units. Now, as this works, the whole contractions, it says, are smooth. The way we see it is when we go past it and then the muscles don't move smoothly. Now, another thing that we need to talk about is that there is this underlying partial contraction of muscles, and it's called muscle tone. Now, there is a disease called Bell's palsy. I don't know if anybody has heard of it or seen it, but it is where half of a person's face is paralyzed. And it's a weird disease. We don't understand it. It can come and go, or it can stay. It's weird. But if you're trying to relax your face, the muscles will still have a underlying muscle tone. And you, again, just like recruitment, you can only really see it when it is no longer there. So for someone who's paralyzed, like with Bell's palsy, their whole face sags. And that is because the muscle tone even is gone. So when we look at how muscles contract, there's different ways that it can happen. I mean, obviously, when we're just talking about muscle contraction, we probably are thinking about, like a bicep, flexing my elbow. I'm going to contract my bicep brachii, and it's going to pull my lower arm up to my head. So the muscle is going to actually shorten. But there are multiple ways this can happen. Now, first of all, isotonic is when muscles contract and they change length. All right? Inside that, it can either shorten or lengthen, which are different. But isotonic is when the muscle contracts and changes length. Now, one way to remember this, and again, I'm not going to say that I'm promoting any of this, but if I am going to drink a gin and tonic, I need to get it to my mouth. So I'm going to have to change the length of that muscle to get my drink to my mouth. So isotonic is about changing in length. Isometric, on the other hand, is where the muscle contracts and it doesn't change length. If I was trying to move a wall or pick up my car, once I got past the give that the shocks have on my car and I try to lift it, I'm not going anywhere. Isometric is when the muscles contract but they don't change length. Now, I always remember this because I grew up in the 70s when they were trying to move everything in America over to the metric system, and nobody liked it. We liked our inches and yards and feet, and so the metric unit got no traction. There was no change, so I could remember that. Now, going back to isotonic, the one that changes length, has two subcategories. Concentric's the one we think of. It's when the muscle contracts and shortens. If I'm working out, that's what I'm doing. But there's also eccentric, when it lengthens. And just think about this. And my example is, if I've got a brand new little baby and I'm going to put that baby down in their crib, I don't just drop them. I gently extend my arms to lay them down. Now, in pretty much every movement, every contraction where there's movement, there will be both concentric and eccentric contractions. They help regulate the movement. So if I am flexing my elbow, my bicep brachia is going to contract concentrically, but my tricep brachii is going to contract eccentrically and help control the movement. Now, in our body, we are going to generally be these up to on the top right, this fast twitch, fatigue resistant fibers. That is our general makeup. Now, I'm going to look at the two on the left side, which are the two polar opposites. So we have slow twitch fibers and fast twitch fibers. If you've been in sports, you might have heard of these. Slow twitch fibers, these are fatigue resistance. They're the marathon runner, basically. They can contract for long periods of time. Now, if they're contracting for long periods of time, they're looking for long-term energy. Long-term energy means I need to get into the mitochondria because that's where all the ATP is formed. If I'm getting into the mitochondria, I need a good supply of oxygen. So if I'm going to have a lot of mitochondria, I need a lot of myoglobin, and I'm going to need a lot of blood vessels, capillary beds, because these are designed to contract for long periods, slow twitch fibers. There are also red fibers. They're called red fibers because they have a lot more blood flowing to them. On the opposite, these fast twitch fibers are also called glycolytic, which tells you that they're only worried about glycolysis. They want immediate energy. They're not worried about long-term stuff, so they're not really concerned about mitochondria. They're going to have a lot less mitochondria inside them. They're going to have a much poorer blood supply because they're not worried about oxygen. They're worried about that immediate first step. Boom, get that instant energy and get done with it. They're the sprinters of this group. Slow twitch are the marathon runners looking for long-term energy. The fast twitch are the sprinters. They just want that immediate burst energy. Now, in humans, like I said, we are more on this one on the right upper, that's the intermediate. But in something like a bird, you can see these two. Chickens don't fly. So their breast meat, the muscles that are needed for flying, are only used for short-term energy. So they are white meat. Their thighs and legs, because chickens run like crazy if you've ever seen them, they are in constant motion and they need much more energy. And so they are dark meat. Slow twitch is going to be dark meat. Fast twitch is light meat or white meat. If you've ever eaten duck breast meat is dark, and it is dark because they fly. So, and this is always fun to talk about around Thanksgiving. If you want to get into this, you can probably get more, if you can deal with it, you can get more food if you kind of gross people out. But anyway, hopefully that makes sense.